It's Biomed Cafe menu. Mitochondrion will be presented today. We will talk about mitochondrion discovery, mitochondrion structure, mitochondrion function, and quiz game. I'm Dr. Biomed, cellular and molecular biologist. Hi everybody, welcome to the Biomed Cafe channel. In this channel, complex biological and medical contents are presented in a simple, classified, and summarized form. Mitochondrion discovery. They saw something others didn't see. Mitochondria were discovered by Albert von Kulinker in 1807 in the voluntary muscles of insects. The term mitochondrion was coined by Carl Bender in 1898. The mitochondrion is popularly nicknamed the powerhouse of the cell by Philip Sienkiewicz in 1957. Mitochondrion characteristics. Unlike other organelles, it has two membranes. It's typically round to oval in shape and range in size from 0.5 to 10 micrometers. It's not visible under the microscope unless they are stained. It has its own genome, called mitogenome. It's found in the cells of most eukaryotes, such as animals, plants, and fungi. Different cell types have different number of mitochondria. For example, mature red blood cells do not have, but liver cells have 2000 mitochondria. Cells with high demand for energy tends to have greater number of mitochondria. For example, heart muscle cells. It has folding to increase surface area, which increases ATP production. It reproduces by binary fission. Mitochondrion structure. Mitochondrion has 5 parts in its structure: outer membrane, intermembrane space, inner membrane, cristae, and matrix. Outer membrane. It's made of phospholipid layers. Protein to phospholipid ratio is 1:1, that is similar to cell membrane. Outer membrane covers the surface of the mitochondrion. Its thick is 60 to 75 angstroms. Small molecules can pass freely through the outer membrane. After disruption of the outer membrane, protein in the intermembrane space leak into the cytosol, then it's leading to cell death. Outer membrane has a lot of contents. Purines. They are integral membrane proteins. They form large number of channels. They play a role in allowing proteins to cross. Another content of outer membrane is voltage-dependent anion channels, or VDAC. They play a role in transport of nucleotides, ions, and metabolites. Another content is translocases. They play a role in transport of larger proteins actively, and enzymes. They play roles in elongation of fatty acids, oxidation of epinephrine, and degradation of tryptophan. Intermembrane space. It's an area between the inner and outer membranes. It's also known as pre-mitochondrial space. Contents of intermembrane space can be divided into two groups: The first group is small molecules, and the second group is large proteins. Small molecules, for example, ions and sugars, that their concentration is the same as in the cytosol because the outer membrane is freely permeable to small molecules. Large protein, for example, cytochrome C, their concentration is different from the cytosol because proteins must have a specific signaling sequence to be transported across the outer membrane. Inner membrane is made of phospholipid. Protein to phospholipid ratio is 3:1. Inner membrane is located inside mitochondrion. It's impermeable to most molecules. It has no purines. It's where most ATP is created, and it has many folds called cristae. Inner membrane has a lot of contents. Proteins that are 151 different ones. Transporters, they play a role in transport of ions and molecules. Translocase of inner membrane TIM complex, they play a role in transport of proteins into the matrix, and enzymes. For electron transport chain, they form a membrane potential across the inner membrane. Function of inner membrane are occurrence of electron transport chain redox reactions, ATP synthase which generates ATP in the matrix, and regulating metabolites passage into and out of the matrix by transport proteins. After disruption of inner membrane, multiple clinical disorders including neurological disorders and cancer occurs. Cristae. These are the folds of the inner membrane. Cristae increase the surface area of the mitochondrion membrane. Then it causes enhancing mitochondrion ability to produce ATP, enhancing mitochondrion ability to take place other chemical reactions, and creating more space for more reactions to occur. They increase the mitochondrial output. There are numerous cristae. In liver cells, the area of the inner membrane is about 5 times as large as that of the outer membrane. In muscle cells, they have a greater demand for ATP, then they have more cristae. Cristernal space. It formed by folding of the inner membrane. Matrix. This is the space inside the inner membrane. It contains about most of the total proteins in a mitochondrion. The matrix is important in the production of ATP. Matrix has a lot of contents. Enzymes. There are hundreds. They play roles in oxidation of pyruvate, oxidation of fatty acids, and citric acid cycle or Krebs cycle. Mitochondrial ribosomes or mitoribosome, that they are 70S, tRNA, and mitochondrial DNA or mtDNA. There are several copies of it. It's more similar to bacterial DNA. Children always received their mtDNA from their mother. Now I want to compare mitochondrial outer membrane with mitochondrial inner membrane. Outer membrane is located at the surface of the mitochondrion, but inner membrane is located inside mitochondrion. outer membrane has purines, but inner membrane has not purines. Protein to phospholipid ratio of outer membrane is 1:1, but protein to phospholipid ratio of inner membrane is 3:1. Outer membrane has not crystal, but inner membrane has crystal. Outer membrane is permeable very much, but inner membrane is permeable very little. And outer membrane has not electron transport chain enzymes, but inner membrane has electron transport chain enzymes. Mitochondrion function. Function number 1, energy production. Energy production takes place on the crystal of the inner membrane. Krebs cycle produces a chemical called NADH in matrix. Then NADH converts to ATP by enzymes of the crystal. In molecule of ATP, energy is stored in the form of chemical bonds. When these chemical bonds are broken, the energy can be used. Function number 2, cell death or apoptosis. As cells become old or broken, mitochondria help decide which cells are destroyed. Mitochondria release cytochrome C. Cytochrome C activates caspase. Caspase involves in destroying cells during apoptosis. Function number 3, storing calcium. Calcium is so critical. Therefore, mitochondria absorb calcium ions and store them until they are needed. Calcium is vital for releasing a neurotransmitter from a nerve cell, muscle function, fertilization, blood clotting, and releasing hormones from endocrine cells. Function number 4, heat production. When they are cold, the body can generate heat by mitochondria of brown fat during a process called leak. Brown fat is found at its highest levels in babies, and slowly levels reduce as we age. Similarities between mitochondria and bacteria. Both have 70S ribosomes. Both have approximately the same size. Both have circular genome. Both have similar enzymes. Both have own translation system. And both reproduce by binary fission. Key points of mitochondrion. Mitochondrion membrane-bound organelle found in the cytoplasm of almost all eukaryotic cells. The primary function of mitochondrion is to generate large amounts of energy in the form of ATP. It has two distinct membranes and a circular genome. It uses aerobic respiration to generate ATP, which is used throughout the cell as a source of chemical energy. Thank you for watching. Please subscribe to this channel, and if you like the contents of this video, please like it. Now it's time to the quiz game. I have put the link to the quiz game in the description of this video. Download it for free and enjoy it. Until the next video, bye!